Hello and welcome everyone to this episode of P&I Tea Time webinar series. Today with Dr. Robert Wawrzinek. He will be speaking about small molecule-based targeted delivery of nanoparticles to the Langerhans cells in human skin. Let me quickly introduce our speaker. So Robert is an organic chemist who obtained his doctoral training from the University of Potsdam, developing fluorescent dyes for bioimaging applications, and after a postdoc in Australia at the University of Queensland, working on organic semiconductors, he joined Christoph Rademacher's group at the Max-Planck-Institute of Colloids and Interfaces in 2016, where he worked on drug delivery systems for immune modulation and cancer vaccination. Just as a heads up, our next Tea Time webinar will be held on the 15th of July by Dr. Tamara Dacoba from the University of Santiago de Compostela. She will be speaking about preclinical development of an HIV nano vaccine candidate from the bench to a pilot plant. And also, we would kindly invite you to our 2020 virtual symposia on July 23rd in the afternoon, speaking about RNA medicine in a pandemic. So stay tuned for the news. And now I would like to hand over to our speaker, Robert.
Well, thanks, Martin, for the invitation and your kind introduction. Good afternoon, everyone. Today, I'm happy to share some of the results we achieved with our Langerhans cell-targeted delivery system, and hopefully convince you of the potential this technology provides for immune modulation therapies. I guess we can all agree that any medical treatment aims to improve a patient's situation by administering a drug to certain target cells. This, however, is often accompanied by undesired side effects and reduced efficacy. This is due to uptake by off-target cells and degradation by the body's clearing mechanisms. By functionalizing your drug or an entity that incorporates it, for instance, with ligands that are only recognized by receptors on your target cells, you can avoid these shortcomings and achieve the desired outcomes in a safer and more efficient way. For the sake of time, I can only highlight a few examples of specific cell targeting, such as delivery of nanoparticles to hepatocytes using galactose or GalNAc ligands interacting with overexpressed ASGPR. Another famous example is the folate receptor, found overexpressed on many cancer cells. The incentive here is also to enhance uptake by employing specific ligands, in this case, derivatives of folic acid. And lastly, a very recent and quite promising approach out of Dan Pier's lab. Their acid technology allows for individually functionalized LNPs to target specific leukocyte subtypes. And this depends on the antibody ligand that is attached to the nanoparticles. But what all these approaches have in common is that they start by identifying target cells. In our case, and the title of the talk already gave it away, those targets are Langerhans cells. They are a subset of dendritic cells and responsible for numerous tasks along the lines of pathogen recognition and processing and elucidation of immune responses. This spans from inflammatory T-cell activation, self-antigen tolerization, through to involvement in B-cell responses in immunity. And those roles are important in infectious diseases, skin-related conditions, and various kinds of cancers. And because of those roles in activation, modulation, and tolerance, we forged the plan to mimic the natural mechanism of pathogen recognition and create a delivery system that can be modularly adapted for individual tasks and indications. The key building blocks comprise of a cargo, which can be a specific antigen one wants to elicit an immune response against, but also toxins and other drugs directly interacting with Langerhans cells. This cargo is encapsulated in a protective vehicle, such as liposomes or LNPs, but can also be extended to PMMA, gold, or other nanoparticles. And finally, and most importantly, the targeting ligand, exclusively recognized by Langerhans cells to avoid off-target effects. Let's have a closer look where to find Langerhans cells, since one additional aspect in designing a delivery system is consideration of the tissue where your target cells reside. In case of Langerhans cells, this is the uppermost layer of the skin, the epidermis. LCs are the only dendritic cells in this layer, and you can see nicely on the right how their dendrons reach out for potential threats, often even touching just below the skin's surface. Now, the mechanism by which LCs deal with pathogens and ultimately our delivery system starts with the receptor-mediated uptake and subsequent migration to the next lymph node. During this journey, the pathogen is processed in endosomal compartments into smaller bits. Those are eventually presented on MHC molecules to prime T cells against any intruder carrying such motifs. But since evolution instructed the whole family of dendritic cells with multiple tasks, including recognition of a variety of glycan structures, it is inherently difficult to target just one subset. First and foremost, for their carbohydrate binding receptors generally exhibit shallow and hydrophilic binding sites, which in turn is of course far from ideal from classic small molecule ligand development. And you can see that there's a plethora of such receptors found on numerous dermal cells, causing potential off-targets. The one that is unique for Langerhans cells, though, is Langerin, and consequently, this was the receptor we were after. It took us a few years, but by constant chemical development and biophysical investigations via, for instance, NMR, we eventually synthesized a ligand that fulfilled the requirements we defined. And as you can see, it is a quite simple carbohydrate-based molecule that can be easily synthesized and covalently bound to other entities, such as this lipid, for example. Now that we have this one central building block, we can combine it with different cargos and vehicles. Today, I will show you liposomes that carry the cargo in and the ligand outside, lipid nanoparticles with encapsulated mRNA encoding for a protein antigen, and even the protein itself, directly decorated with a ligand on its periphery. All these variations we call LC-TDS, the Langerhans cell targeted delivery system. Now let's have a look what they can do. So first off, we prepared empty liposomes without any cargo. To trace them, we added a fluorescent dye as part of the membrane and tested them for selective binding and uptake to Langerin using several cell lines expressing different C-type lectins. And indeed, we confirmed a high selectivity for Langerin in comparison to wild-type, DC-SINE, dectin, and other overexpressed lectins. In this video, you can see rapid uptake and continuing accumulation of the red-colored liposomes. The reason for this accumulation is that the ligand only temporarily binds to Langerin. After uptake, the liposomes are released within the endosomal compartments so that the receptor can recycle back to the cell surface for yet another round. And this is one of the key advantages the LC-TDS has over, for instance, antibodies, which, due to their very strong affinity, prevent more than one uptake event per receptor. Next, we looked at primary Langerhans cells, which we obtained from fresh human skin explants. From those, we remove the excess fat tissue, and after a digestion step, we can peel off the epidermis from the dermis, which is then further processed to create cell suspensions. After exposure to the targeted liposomes and by using flow cytometry, we confirmed that we can actually target more than 97% of all available Langerhans cells and way below 0.1% of other bystander cells. Here it needs to be stressed that LCs only make up for around and about 2% of all cells in the epidermis. The majority are keratinocytes, which, by the way, themselves express carbohydrate recognizing receptors and are therefore considered potential off-targets to avoid. The next example shows LC-TDS LNPs, again labeled with a dye and filled with Cy5 labeled mRNA encoding for GFP. In this video, you can see live cell imaging on Langerin expressing HEK cells over 14 hours in the 3 respective channels. Nicely, the mRNA and liposomes are co-localized, and after roughly 9 hours, the GFP expression joins in, showing the successful mRNA delivery and protein expression. Now, when we look at flow cytometry data, we can see that for LNPs that carry the mRNA but are lacking the targeting ligand, we observe a fair amount of unspecific uptake into Langerin-expressing cells. This is expected since almost all LNP-based therapies and vaccination approaches are based on this unspecific uptake. But what you can also see is little to no GFP expression. In fact, this is only observed when the LNPs are equipped with the ligand. So why is that? In order to have efficient mRNA protein translation after the uptake, the mRNA has to be quickly released out of the endosome into the cytosol. We think that the mechanism our targeting ligand follows is inherently different from the unspecific uptake and actually promotes endosomal escape and consequently robust protein expression. Adding another level of complexity, we teamed up with our dear collaborators from Innsbruck and encapsulated EBNA1 into targeted liposomes. EBNA1 is an antigen of the Epstein-Barr virus, and our aim was to elicit an antigen-specific immune response. Now, EBV is a neat human system for this, since roughly 95% of the population are positive for this virus. So we exposed our liposomes to LCs from 2 donors, 1 positive and 1 negative for EBV, and subsequently co-cultured the LCs with T cells from autologous blood of the respective donor. And you can see here, for the positive donor, the significantly higher cytokine response from CD4 positive T cells against our EBNA1 loaded and targeted liposomes. Untargeted liposomes, however, did not cause a profound effect in any of the 2 samples. A similar setup was used in mice in collaboration with the Helmholtz Center for Infectious Diseases in Braunschweig. Here we used ovalbumin as an antigen and not only prepared liposomes, but also directly functionalized protein. At this point, I have to explain that for mice experiments, special transgenic animals are required. The reason for this is that our ligand is not only highly specific for Langerin, but in particular for the human receptor. Therefore, we have to use these transgenic mice expressing the human Langerin on their Langerhans cells. So we took LCs from those animals, exposed them to overloaded liposomes or the targeted protein, and subsequently to overspecific T cells from OT12 mice. And here we observed a pronounced T cell proliferation for both targeted LC-TDS variants, the encapsulated and the directly functionalized ova. Now that we have shown in cell suspension experiments that our system works as intended, we also had a look at the administration route, because of course it doesn't help your cell-specific delivery system being deposited remote from your target tissue. When it comes to skin as a target tissue, the first that pops into mind is probably conventional injection. But since LCs are exclusively in the epidermis, even a transdermal injection potentially deposits the LC-TDS way below that layer, wasting a lot of material that cannot be reached by LCs. Therefore, we have been investigating alternative technologies that fulfill the requirements of low penetration depth. Those are microneedle patches, microneedle injection devices, and the transfollicular administration, which exploits access to LCs at the end of hair follicles. But for the sake of time, I will only talk about the first 3 of those options. First, we tested conventional intradermal injection. For its small size and therefore comparatively good mobility, we chose targeted GFP protein and injected it ex vivo into human skin. After isolating the epidermal LCs, we could confirm to have reached more than 95% of available LCs in the injection site, whereas untargeted GFP was not found in the Langerhans cells. In collaboration with Ryan Donnelly's group from Queen's University Belfast, we explored microneedle patches. They consist of polymers in which liposomes can be blended in during their casting process. With 600 micrometers in length, their needles are just long enough to penetrate the stratum corneum and set themselves into the dermis. The stratum corneum is the cornified top layer of the epidermis and is a major barrier when it comes to skin penetration. Due to their conical shape, most of the microneedle's volume is actually placed in the epidermis. There they can eventually dissolve and release the previously embedded liposomes, which are then ready for uptake by LCs. Here too, we found almost all LCs positive for the LC-TDS, with less than 0.01% of targets. Non-targeted liposomes, in comparison, only reached a mere 0.1% of LCs. The last example I want to show you today is the use of microneedle injection tips from nanopads, which conveniently can be mounted to conventional syringes. Their main deposition depth is around 600 micrometers, so below the epidermis, which means that LC targeting depends on vertical diffusion of particles from the dermis up into the epidermis. For proteins are still the smallest kinds of LC-TDS, we again first injected targeted GFP and were delighted to find in the subsequently obtained epidermal skin suspensions 90% LC-TDS positive Langerhans cells. I have to note that the results I show you on this slide are preliminary and in part only a few days old. But since they mark an important milestone for us, I still want to share our excitement with you. So for the last few weeks, we have been testing LNPs in conjunction with the nanopads devices, though still without any mRNA as cargo. But after a few disappointing tries, we learned that LNPs larger than 100 nanometers were not able to diffuse into the epidermis. So we changed our formulation accordingly to obtain smaller particles. We also optimized the administration process by moving from one single injection to several little ones distributed over a larger area. We think this reduces the pressure in the injection blebs, preventing the LNP solution to be pushed further down into the tissue. As opposed to the previous skin experiments, we this time also had a Zimmer dermatome at hand, which enabled us to detach the desired skin layers in seconds rather than, well, hours. As a result, we finally achieved for the first time perfect LC targeting with LNPs in human skin, which now blazes the trail to add mRNA to the equation, hopefully showing the first mRNA-mediated protein expression in primary Langerhans cells. We are also preparing in vivo experiments on our transgenic mice with this setup, hopefully starting early next month. To conclude, I hope to have convinced you that our LC-TDS is an elegant drug delivery system with a huge potential for immune modulation via Langerhans cells. It is granted access to those by recognition of Langerin and shows highly specific uptake in cell lines and primary cells. Due to its modularity, we were able to deliver all sorts of targeted nanoparticles and could trigger antigen-specific T-cell activation in men and mice, as well as antibody responses in vivo, which I did not show you today. And lastly, I also wanted to convey that the administration route is also of profound importance to fully use the efficiency advantage targeted drug delivery systems provide. I want to end by expressing my gratitude to have the privilege working in Christoph Rademacher's group, who recruited a bunch of talented and amazing people around him. In context of this talk, namely Mareike, Novras, Dongyun, and Felix. Special thanks, of course, go out to our fantastic collaborators and Peter Seeberger's department within our institute. Martin and Jürgen from Precision Nanosystems, as well as the people from Nanopass and Zimmer Biomed for their support. With that, I also want to thank you for your attention, and I'm happy to take questions.
Well, thank you very much, Robert, for that. Very much appreciate you giving that very interesting talk. So very much appreciated it. Really nice data there. So always nice to see visualized by videos as well as talking. So really good to see those there. So all that remains for me to say is thank you very much once again for giving this very interesting talk. We really do appreciate it. The next
pleasure was mine.
Yep, the next Tea Time webinar will be in 2 weeks' time, and we will be sending out details of that. And if there are any questions we haven't been able to answer live, we will follow up, and the video of this presentation will be available in due course as well. So thanks everyone, and particularly thanks Robert.
Thank you everyone. Bye bye.